So before we get into talking about special senses, for those that took a look at the autonomic nervous system, were there any questions that came up when you guys were reviewing that? Something that needed to be clarified? Anything that didn't make sense to you? Yeah, go ahead. Oh, the tonic firing, yeah. Okay, so actually it turns out in a few instances that there is some steady firing of the neuron to stimulate the effector, right? And so the example you're getting at is how you can have opposing effects from the single innervation, the sympathetic nervous system. The example I was showing you guys is of the blood vessels. So let me bring up that slide. And it was the sympathetic nervous system that was involved with that. Let's see, there we go. So this one where you had opposite effects without dual innervation. Because remember, the sympathetic, parasympathetic, a lot of times they antagonize each other. One's doing one, one's doing the opposite. In this case, what we have is you have innervation and there's tonic firing, meaning that there's some steady rate firing of the particular neuron, in this case a sympathetic neuron, on blood vessels. Okay, certain types of blood vessels. And so that means that there's some state of contraction. It's not fully contracted, but there's some state of contraction of the smooth muscle that's in the blood vessel walls. Everybody with me so far on that? Okay? So if we want to decrease the diameter, are we going to increase contraction or decrease it? So if we want to decrease diameter, so we're going to increase the contraction of the muscles in this? Yes, right? We're going to increase contraction, that's going to decrease the diameter. How do we do that? Well, that happens by increasing the firing frequency, right? So we start off as some intermediate frequency, and we increase it. That's going to cause those smooth muscles to fire even more, contract even more, causing that blood vessel diameter to decrease in diameter, right? Everybody with me so far on that? Now, if we have tonic firing, what if we reduce the firing rate? So frequency becomes even less. What's going to happen to the diameter of the blood vessel? Is it going to decrease or increase? So if we decrease the firing, so the diameter should increase, right? So that means that the smooth muscle that's in the blood vessel walls, right, are they becoming more relaxed or more contracted in that case? More relaxed, right? So we start off with some level of firing. If we increase it, more of those smooth muscles are going to contract, or they're going to contract even more, decreasing diameter. Now, if we start off with the same tonic firing, and then we decrease that firing rate, that means that even fewer, or that contraction of smooth muscles is going to decrease, which means that blood vessel diameter is going to be elevated, right? It's going to increase. And so that's what we're talking about. We're able to control the diameter of certain blood vessels by having just a single branch of that autonomic nervous system innervating. Does that make sense? Yeah, awesome. Everybody with me on that? Okay, good. Any other questions on the autonomic nervous system for those that went over it? Things that were unclear, fuzzy to you guys? No? Okay. Well, if you guys do go over it and things are, become unclear or not making sense, be sure to stop by my office, so we can clear things up, right? So I don't want you guys to fall behind because, you're stuck on something that doesn't make sense to you. All right? Okay. Okay, so on to the wonderful world of special senses. So up to this point, we've talked a little bit about senses and sensory receptors as receptors for viscera, for stretch, for monitoring temperature, also feel and touch and texture and so forth. The special senses, they're grouped specially because they're specialized organs that house special types of sensory receptors. In a lot of cases, those are specialized neurons. And so we're going to be taking a look at these individually. The first ones that we're going to look at, though, are really coupled together. Olfaction and gustation, so taste and smelling, right? They're kind of really closely related, all right? And so here we're going to take a look at some of these. And in the regular theme for looking at these specialized sensors, or special senses with these special organs, is first talk about just enough anatomy so that we're on the same page, and then we'll start talking a little bit about mechanism, the physiology behind what's going on. And now that you guys are experts in knowing all the different regions of the brain, we'll talk about, where these things are routed towards the brain, right? So here with olfaction, we have to talk about, all right, well, we have that olfactory bulb. You guys saw that in the sheep brain dissection you all did, right? And projecting from the olfactory bulb is you have some olfactory neurons whose dendrites are actually specialized so that they have olfactory receptors on them. Now, each olfactory receptor is going to be triggered by a particular chemical, olfactant. All right, so here, this is one of the few places in our body that our nervous system is in direct contact with the external environment. Right? These olfactory neurons have sensors that are projecting into the external environment. Right? And so here, these olfactory, endings, all right, have those olfactory receptors on them, which means that they bind a particular chemical and that sends a signal, right, along the olfactory neuron where you get synapsing, and it further is going to be routed to areas of our brain for processing and then eventually interpretation, right? So you guys can probably anticipate some smells kind of give you a really emotional response, like, ew, I do not want to go there, right? Or, wow, that really smells good. That must be a Krispy Kreme donut shop over there, right? So what part of the brain are some of these tracks heading to if it's giving you an emotional response? So what type of system? The limbic system, yeah, so you guys are spot on, right? So part of what gets routed, the routing is going to be towards the limbic system here, all right? So you guys already anticipate, some of what we're going to talk about when we talk about smell. All right, so how do we smell? Well, we have some olfactants that are going to be coming in, and they get churned up a little bit by some of the, features that are in the nasal cavity. And so here, just to give you guys a orientation, this is a zoomed in portion of right in that area. So here is a bone, and this is going to be the olfactory neurons coming through the bone. And then you have the dendrites sticking out into the nasal cavity. And so remember, this is generally going to be embedded somewhat in mucus. And you're going to have these dendrites have the olfactory receptors that bind to an olfactant, and they send that signal back up. Now, does anybody know what bone this is? So it's one of the funny sounding bones that was kind of interior, kind of back. It's anterior to the sphenoid. So what's that other one that sounds like the sphenoid? Ethmoid, so E before S, as you go anterior to posterior. So it turns out these guys project through the ethmoid bone. So if you guys take a look at the ethmoid bone, you can see little holes in it as part of the cribriform plate, and that's where the olfactory neurons are going in. So if someone has a massive impact that fractures the ethmoid bone, right, which sense is going to be affected? Sense of smell, right, olfaction. Now, why is that dangerous? Well, think about natural gas, right? What do they put in natural gas so you guys can smell? Because it's normally odorless, right? Yeah, so it's kind of the sulfur type of smell. So if someone has a problem with smelling, right, that could be really dangerous. Okay, so here what ends up happening is that we have odorant molecules that are going to be taken into the nose, and they're going to be kind of churned around by the features in that inside of our nasal cavity, right? So we have conchi that are there that kind of churn the, mix up those air molecules that contain the odorants, so turbinates. And then they attach the olfactory receptors that, remember, are on the ends of these olfactory neurons. And when a olfactant binds to its cognate receptor, meaning that that's a receptor that will recognize that olfactant, it is going to generate a graded potential that can be an action potential if that graded potential is large enough. So does that mean that this receptor activation, is that excitatory or inhibitory? If it's going to generate an action potential, potentially. Excitatory, absolutely. Right? So here we can get an action potential that is going to propagate down the axon, passed off via synapse to another neuron that's going to take it to various areas of the brain. Like you guys said, one of those areas is going to be part of the limbic system, right? Because there's an emotional response here. Right? Does the sequence of events make sense to folks? And then the anatomical features that are going from one sequence of that event to the next. Everybody with me on that? All right. So anybody remember which cranial nerve is involved with olfaction? Is it number 12? Number 10? The vagus nerve? Definitely not, right? So remember I was telling you guys that generally we go 1 through 12 as we go from front towards the brainstem, right? So here, cranial nerve, number 1. So olfactory nerve, right? And so it gets routed here. Some of these are going into areas of the limbic system, right? All right, so processing smell. So we do have an area, right, where we're consciously aware of that certain smell. And so that is going to be here with the olfactory cortex. It's kind of in that area that leads to the insula. So that lateral sulcus there, right? It's part of the temporal lobe. And remember, with primary sensory areas, that's where the sensory information first gets routed for processing. And then to be able to identify and interpret them, to say that is a donut or that is a carrot, that's being done in a sensory association area, right? So here, we have our olfactory cortex, right? And then, and then we're going to have a olfactory association area, which isn't on here. It's going to be in the same area, okay, that is going to be doing the identifying and interpreting of that, right? Now, the pathway, there's 2 separate pathways, right? One of those pathways, though, from the sensory information gets routed to the limbic system. The other part for our conscious, knowing what that is going to the cerebral cortex, these association and primary areas. And that reminds me, I posted up on our Blackboard links to a really good documentary by Nova on secrets of the mind. And in there, it has a variety of, things that happen to the mind, such as, individuals that are legally blind, but they can still determine whether things are going up, down, left, right. So it's called blind sight. And if you guys are interested in taking a look at that, the link is posted up on Blackboard. There's other things in there, like the Capgras syndrome, where people think that individuals are replicas of the people they know, right? And so it's fascinating. I encourage people to take a look at, that documentary. All right? Okay. So processing smell is closely linked with gustation. Or I should say, a lot of what we taste is actually smell. But we do have taste buds that are localized, generally, not so much in the center of the tongue, in the middle area. And I'll show you kind of distribution where they're more concentrated in just a second. But our taste buds, if we zoom in on them, right? If we zoom in really tightly to a taste bud, you can see that here is going to be our neurons, our gustation neurons, but these aren't in direct contact with the external environment. So here, one of the themes for some of these specialized organs, the sensory neurons, there's a special sensory receptor cell that sends the information to a sensory neuron. So it's one of the differences between olfactory neurons. The olfactory neurons, the dendrites themselves with the receptors, are sticking into the external environment. That's not happening for these guys. And so here we have, with the taste buds, you have microvilli that helps filter in molecules to these receptor cells that are going to send that information through the synapse here, okay, is going to excite the gustatory neuron, and then that neuron is sending information along its axon, along the gustatory tract, to different parts of our brain for interpretation and identification. All right? And so there's different receptors for different types of taste. Okay? So here, we're going to have perhaps some different areas of our tongue are going to have different concentrations of these receptors. So this is a taste pore that's part of a taste bud. Okay. Everybody with me on that? Okay. So there's generally considered 5 different types of taste receptors. And so we got sweet, sour, salty, bitter, and something called umami. There's the 5 basic types of taste receptors. Their distribution, where they're concentrated, are a little bit different. So it turns out sweet's concentrated a little bit more with the taste buds that are on the tip of our tongue. Salty is a little bit broader, extends a little bit further to, lateral portions of the tongue. Sour is a bit on the sides of the tongue. It's not to say that these are just areas where these receptors are more concentrated. It doesn't mean that they're only found there. All right? Bitter's in the back of the tongue, and then umami is a little bit more broadly distributed. Now, as you can see, most of these are G protein coupled receptors. These other 2, sour and salty, we'll talk about in just a second as well. Sweet, I think everybody kind of understands what sweet is, right? Sour are involved with, say, acidic foods, right? So acids, like in lemon, citrus fruits, tomatoes are fairly acidic, and so forth. Salty is going to be measuring sodium chloride or other types of metal ions, and so that's that one. Bitter, so it turns out bitter, a lot of what's bitter comes from plant alkaloids, and those are concentrated at the back of the tongue, and so they're intimately involved with the gag reflex, and we'll talk about why that is in just a second. And the last one is something called umami, which is triggered by certain amino acids. And remember, amino acids make up proteins. In particular, the amino acid that triggers this receptor particularly well is glutamate. So this is why monosodium glutamate is added to certain foods, is to trigger the umami taste buds, or the umami receptors, right? Okay. So if we take a look at some of these gustatory cells, the ones that have a lot of the salt or sour channels, receptors, they're actually channels. And so here, if we have, say, sodium chloride, that is going to be going from outside the cell to inside of the cell, what is that doing to the membrane potential there? We're changing the membrane potential, right? So if we add positive charges to inside a cell, is that going to be making the membrane potential less negative or more negative? Less negative. So it's more likely to generate a action potential, right? So here, this whole channel or receptor is actually just a channel, right? You throw salt on your tongue, what ends up happening is that you're depolarizing, right, that part of the tongue, and therefore potentially activating the receptor to send that signal, right? It's depolarized, and then you get neurotransmission to the gustatory neurons. Same thing with sour, so acidic foods. So remember, acidity is really going to be hydrogen ions or protons, and they do something similar, right? So here we don't have any G protein coupled receptor. It's just a channel that's allowing passage of substances that are part of our food intake into the cell. And if they are going through this channel, they're causing depolarization, and then that is causing the signal to be sent. And if neurons that are coupled to these receptors fire, the brain knows that it's a sour sensation or it's a salty sensation, right? So it's mapped to the receptor. Everybody with me on that? Okay. Just like with some of these others, we have a G protein coupled receptor, which you guys know about, where we have the receptor, and then we have our inactive G protein. And so if it's inactive, would it be having GDP or GTP bound to it? GDP, right? And then if we have that molecule binds to the receptor, and this G protein is now active, it would have what? GTP bound to it, right? And then you get second messengers to cause a signal to be sent along that gustatory neuron. And when that gustatory neuron is sending that signal, it's mapped to either the umami, the bitter, or the sweet, pathway. So I'll tell you something that's really interesting. One of the guys who is studying these taste receptors in mice, what he ended up doing is he decided, well, what if I take, say, the bitter G protein coupled, just a receptor portion, and I swapped it with the sweet. So he swapped the two. And so now these mice, when they were given something bitter, it was signaling via the sweet pathway, right? So they thought they were getting something sweet. And so when you give them something bitter, they were constantly thinking, wow, that's really sweet. And they got more and more of it, right? So they swapped the receptors. It's pretty crazy. Now, I told you I was going to get back to talking about the bitter receptors. Turns out the bitter receptors are much more sensitive than the other receptors. And I also told you that the bitter receptors, a lot of what triggers the bitter receptors are plant alkaloids. Now, a lot of plant alkaloids are poisons. So why would you want to have increased sensitivity, you think, to that bitter taste? Yes, you can be much more sensitive to it, therefore you can detect lower concentrations before it causes you harm. Why might you want to have the bitter sensation tightly associated with a gag reflex? Yeah, so you get it out of your body, right? Okay? Now, I always wondered, when they were trying to figure out whether poisons tasted bitter or not, how did they get people into that study, right? One of the graduate students say, I'm going to give you poisons, you tell me what they taste like. Who signs up for that, right? So I always thought that was interesting. Now, hopefully they just gave them a little bit, right? But it's an interesting conundrum. How do you figure this out? Okay, so bitter receptors, they detect in really minute amounts for the reasons that we were just talking about, right? The other four are a little bit less sensitive, and the tongue is innervated by kind of three different cranial nerves: the facial, glossopharyngeal, and the vagus, right? And so here, those taste sensations are routed to the gustatory cortex, which is located in the insula. So if we take a kind of coronal or frontal section of the brain, and this is the lateral sulcus, and so here is going to be our parietal lobe, there's our temporal lobe. All right, if we look between that sulcus there, we can see the insula. Okay, that's where the gustatory cortex is. Right? So here's the facial, and then glossopharyngeal, so you can see that this one is innervating. The pharynx is back of the throat here, and so that's maybe one of the ways you can remember the glossopharyngeal. And then the last one is the vagus nerve, which is innervating kind of the lower part of the throat there. Does that make sense to folks? So it turns out that if you hold your nose, though, you have difficulty in tasting. And one of the reasons is because the information that's coming via the gustatory receptors is being integrated with information coming from the olfactory receptors as well. All right. So what ends up happening? Well, first we are going to ingest food and then break it down into some smaller pieces, right? So mastication, it mixes with saliva, and we're going to kind of make those molecules, those taste molecules, a little bit more, readily dissolvable so that they can trigger receptors located in that taste pore. And so the food molecules enter the taste pore, and then they're going to trigger those individual gustatory cells that have certain types of taste receptors that we talk about, right? So just like with the olfactory receptors, because they're triggered by chemicals, most of them are, they're called chemoreceptors. Everybody with me on that? Okay. And so because they are leading to a possible action potential, be routed to the brain for interpreting and identifying. Okay? This is depolarization, meaning that these are excitatory events that are happening. Right? Does that make sense to folks? Kind of the similarities for the pathway here. One of the major anatomical differences between this and the olfactory neurons is that the olfactory neurons themselves have the receptors. Here we have a cell that has the receptors that passes information onto the gustatory neurons that further relays that information to the insula where the gustatory cortex is located. All right. So, like I said, about 80% of taste is smell. And you can even try this out. Though there are other receptors on the tongue. So there's not just gustatory receptors on the tongue. So there's receptors there for texture. So there's certain cultures that like the texture of slimy food. That's not just necessarily popular here in Western culture, particularly in the U.S. But there's some cultures that like that particular texture, right? There's touch and pressure, temperature, and there's also pain receptors that influence the taste experience. So there's receptors that are triggered when you eat something that's really, really has a lot of heat with it, right? So that molecule that activates those receptors is called capsaicin. It's the same thing that gets that can cause, pain receptors to be triggered and heat to be triggered on your skin. And it's concentrated in certain types of foods, peppers and so forth. All right? There's some drugs that interfere with this. So chemotherapeutics, there's several chemotherapeutics out there that interfere with gustation. And so individuals that are on certain types of chemotherapeutics for cancer have problems with, eating food because it doesn't taste very good. All right? Yeah. No, that's separate. It actually physically, interferes with the pathway for the gustation. Yeah. It could be other molecules in that tobacco, that's affecting it. I don't know too much of those other molecules. Yeah. The nicotine may interfere as well, with certain receptors. If you overstimulate them, they may downregulate or they may be damaged. That's my guess. But yeah, it's a really good question. Yeah. All right. So like I said, we'll get a little bit into vision here, and then we'll finish up with vision and then hearing and equilibrium on Thursday. Now, before we get into vision, does gustation and olfaction, is that making sense to folks? All right. Any questions on that before we move on? All right, so we're going to start off kind of like we did for olfaction and gustation with talk about some anatomical features. And then we'll talk about some of the physiology for vision. So what we call structures that aren't involved directly in vision, but are involved with or help with protecting the eye, the major organ there for vision, are called accessory structures. So we have the eyebrows, and then we have eyelids, which are palpebrae, is what they're called. And we have certain muscles that allow us to control those palpebrae. And then we also have orbicularis oculi that are also involved with being able to control muscles or of these accessory structures around the eye. Now, the movement of the eyeball itself is done by these extraocular muscles. Okay? So it turns out that in the lab, you can see this model where there's muscles on the top, the bottom of the eye, and on the sides. And then there's some muscles that are kind of at a diagonal. That one is going to be a abducens, one's going to be the trochlear. And the reason there's some diagonal eye muscles is because the superior and the inferior extraocular muscles are actually attaching slightly at an angle. So if they contract, the eyeball tends to rotate out or rotate down. Right? And to offset that, you have some diagonal muscles that help to offset that, to correct for that. All right, and so what controls these muscles? Most of them are controlled by the oculomotor nerve, which is cranial nerve number 3. The other ones are the abducens and the trochlear. So when you abduct something, is that going towards the midline or away from the midline? Away, right? So abduction. And so here, this muscle slightly rotates the eye away. So it helps compensate for the inward pull of the eye from some of these other muscles. And then we have the trochlear that does something similar. So this word trochlear, in Latin, trochlea is a pulley. You guys take a look at this muscle in the eye. It runs along the medial part of the eye, and then it curls around, kind of a pulley, and then attaches to the eyeball. So that's where it gets its name. All right? So you can't quite see this on here. All right, some other structures that we have. So those were the extraocular muscles. On the eye itself, there's a membrane that covers the anterior part of the eye. It's called the conjunctiva. And so you can kind of see it here. And it covers this part of the eye up to where we start having the cornea. And then it loops back around on the interior side of the palpebrae. Everybody see that? So it kind of does this complicated thing where it loops back around. Okay? So it's a clear membrane. If this membrane, there's an inflammation, if it gets inflamed, it's called conjunctivitis, pink eye. This happens when there's an infection, for instance. The very anterior part of the eye is made up of the cornea. Very anterior part of the eye. Okay. So that's the conjunctiva. So let's do the tear production, what's called the lacrimal apparatus. So the lacrimal apparatus, so here's where the nose would be. Okay? The lacrimal gland is located in this area, and it secretes its secretions over the top of the eye, and they wash down in this direction. There's two exit points for those lacrimal secretions that are called lacrimal puncta, which lead to this lacrimal sac, and then the nasolacrimal duct, which empties into the nasal cavity. All right, so here, the general flow starts here, washes along to here. So how can we overflow the lacrimal secretions? Well, what's one way that can happen? So tear production. So how can we have this so that we generate, or we're having more fluid here that can be than what can be removed? So what's some ways that can happen? Watching. Very nice. So basically, emotional response to increase output from the lacrimal gland, right? To the point where it overwhelms the ability of these lacrimal ducts to remove that lacrimal secretion, right? What's another way if we don't change the lacrimal gland output? Yeah, that's it. Yeah, so if you clog the puncta, which are really, really tiny, all right, or if you clog the ducts, right, then you can also cause tearing. Absolutely. Those are the two major ways. Absolutely. Sorry, go ahead, Logan. Mm-hmm. Yes, absolutely. Yeah, so you're just overwhelming. So like kids, when they're crying and they have the sniffles, right? One of the reasons is they are pouring those lacrimal secretions into the nasal cavity. And they're generating so much lacrimal secretions that it's overwhelming the ability of the eye to hold them as well. So you're kind of flooding the nasal cavity and you're overflowing the eye. Yeah, good question. Any other questions? Yeah. So, you may be able to see some of the lacrimal puncta if you look really closely. They're really tiny red dots. All right? So this is a cross-section of the eye where you have the conjunctiva, you can't see here, but you have the cornea. And then this is the pupil, and on each side of the pupil is the iris, where we have muscles that allow increasing the diameter, decreasing the diameter. The pupil is just an opening between the iris, right? So we also have something called a lens, which we'll talk about. It's important for the focusing. And we have two places where we have a liquid. One place is over here where we have the aqueous humor. So humors are liquids, right, or fluids. And so this is the anterior chamber with the aqueous humor. The other one, which is really big here, is the vitreous humor in this vitreous chamber. One of the things that these liquids do is to help keep the pressure inside the eye, pushing some of these structures, making sure that they're properly pushed to the external portion of the eye. If that pressure is disrupted, you can have vision problems, and we'll talk about one of those in just a second. Now, one of the things is, here the cornea is avascular. So what does that mean? It's avascular. It doesn't have blood vessels, right? So does that mean the immune system can get to it? No. So one of the things is that you can have corneal transplants, and that transplant isn't going to be rejected because the recipient, that area, isn't going to be accessed by the immune system, right? So corneal transplants, you don't have to find necessarily a proper match because the immune system isn't going to be identifying it as a foreign object. Okay? So it's one of the reasons that corneal transplants can happen without having an exact match. All right? Now, I want to show also some of these other things that we have going on here. There's 3 major layers of the eye, where the outermost layer is called the sclera. You'll see this in the lab. That's that white portion of the eye that you see. And it's really tough. And one of the reasons it's really tough is because the extraocular muscles have to attach to it. So if it wasn't tough, when the extraocular muscles contract, your eyeball wouldn't move. It may tear off that tissue. Underneath that, we have something called the choroid. Now the choroid, if you track this all the way back to the anterior of the eye, is continuous with the iris, okay? And then also what we call ciliary muscles that are important for changing the thickness of the lens. All right, so the choroid also has a lot of the blood vessels. And it's fairly dark, so that when light comes in, if this was like a mirror, it would bounce all around, right? Okay, so the choroid's fairly dark, so that when light comes in, when you have an image that comes in, right, it stays focused. You don't have those light, waves bouncing from one side of the eye to the other, making it difficult for vision. The last layer, here the internalmost layer, is called the retina. This is where we have the sensory information, right? The sensory neurons that are detecting photons, light particles. And when they get activated, they're sending signals along their axons that exit here to the optic nerve. All right, everybody with me on that? Okay, so this I think this is one of the last slides. So here, if we take a look at the back of the eye, we're taking a look at the retina, and you can see kind of the choroid behind it with all the blood vessels, right? There's the optic disc or optic nerve. And if you look at the previous slide, you'll notice that the retina stops here, and the retina stops here. And that all we have are axons exiting here, which means that there's no sensory information that can be captured here. So it's a blind spot in our eye. It gets filled in kind of by some sophisticated brain functions. So where we have the optic nerve exiting is actually this optic disc here. Okay? Now, there's a portion of our eye where we have higher concentrations of, high resolution eye receptors. And so this is called the macula lutea. And then even within that, so if you think of this as kind of a target, the fovea is the bullseye. Okay? And this has a really almost exclusively high concentration of what we call cone receptors, which are high resolution, but also they are doing color. All right? Now, how many folks have seen those retinal scanners in order to get into like really sophisticated banks or something like that, right? Okay, so what that is doing is that each individual person's eye has a unique signature of blood vessels. And so what it's doing is using that kind of like a fingerprint. Okay, so not two eyeballs back of someone's eyeballs have the same, pattern of blood vessels. So it's a retinal scan. And basically what it's doing is just matching, okay, you've said that this retinal scan is you. I will match this retinal scan to see if it's you. That's what it's doing. Okay? All right. Sorry. Go ahead, Moses. Well, eyesight, we'll talk about refractive disorders. So some people are near or farsighted, right? And so we'll talk about that, and also astigmatism. You guys will see that in the lab as well. There are some genetic variations that do go on, but there's other disorders that infringe upon eyesight. Good question. Okay. All right. So does this make sense? Optic disc, the retina, the fovea, and the macula lutea. Is that making sense to folks? You guys will see this in the lab as well. All right? Okay. All right. So I think we'll end there.